and blue water. I don't know, you can come up with anything.
One of my friends in there, she also, if it helps you, she made a recipe. Wrote like a recipe card, so she remembered it like a cake mix or a.
Yeah, I would definitely like make little cards. That's what I did when I made a card of the Graham staining technique, and then on this card I have all 4 steps. So when I was studying for the lab, I would go over it in my head, and then I'd check my answers. So that's what helped me. I don't know if you guys are, or go online and see if you can find a video if you're more visual like that. Okay, any questions on acid-fast staining? Okay. Oh, and the clinical significance of this, of course, is if somebody comes in and the doctor orders an acid-fast stain and it stains positive, they could have either tuberculosis or leprosy. So just remember that.
Didn't he ask a question, something like, the test came back blue, can you say that
Right.
this is?
Yeah, so he'll kind of ask you a question in a way that you need to interpret the results so that you show that you really understand it. So if he orders the culture and then you find out that it stains blue, can he begin treatment for leprosy or tuberculosis?
Well, can he get a positive, is that positive that he has tuberculosis?
Right, or some, yeah.
Yeah.
And it would be no, because acid-fast blue is negative. Okay. And again, he might have a slide and say, this bacteria has been stained acid-fast. Is it acid-fast positive or negative? So you'll just be able to, if it's red, it's positive. Blue, negative. I know we're saying it a lot, but it's just one of those things that you kind of need to get down. Because he's going to ask something about either Graham staining or acid fasting. So. Okay, so I'm going to move on to lab 11, which is the streak plating technique. So you guys should all be able to do this. On the test, he might have 4 Petri dishes, like pictures, and you might have to draw it, like the actual technique. And it'll be fun. Just remember that you have to flame the loop in between each step. Because we had somebody in our lab that didn't do that, and it was just like, there was just bacteria growing all over the plate. So, yeah, you have to remember to sterilize the loop. And so you just pull a little bit from here, a little bit from here, a little bit from here. And if you remember from your questions, there's not always going to be bacteria in the 4th sector. I mean, it just depends on how much bacteria you start out with.
I have a question.
Yeah.
In his notes, he actually puts as 3 sections, and then in the lab, it says 4.
There's definitely 4 in the
There was
lab.
okay.
There's the primary.
Sorry, I'll make sure.
Yeah, and then you turn it and do another one, turn it, do another one. And do.
Okay, so make sure there's 4 when he has it.
Yeah. So as far as the lab is concerned, you do it 4 times. And yes?
Is it does it matter if it's clockwise or the other way?
No, I don't think it matters. You just turn it. Yeah, it just says turn the plate. In the book, it looks like it's counterclockwise, but
Okay.
I wouldn't worry about that at all. Okay, so why do we inoculate the loop after each step? It's so that you can get a pure culture. What is the purpose of the streak plate technique? The purpose is to isolate bacteria. Like single colonies will arise from just one bacteria, and then you can just view that colony and kind of see what it looks like and that kind of thing. Okay, and let me see. What are some advantages and disadvantages of the streak plate technique? I actually only know one of each. So the advantage is that you can get even if you start with a mixed culture, you can get a pure culture of bacteria. The disadvantage is that you can't count the amount of bacteria in the original sample. In the lab book, there's the pour plate technique, and that's the one that you use to count bacteria. But just know that the streak plate technique just isolates bacteria. Okay. So any questions on that? Just make sure you know how to draw it. Okay. Wow, so I think that's all the labs that you guys have done so far. These next 3 are the ones that you're going to go into lab and do today. Do you guys want to go over these ones? Okay. These ones are a little bit more challenging than the earlier labs, I would say, especially lab 14, because there's 3 different tests in that lab. But we'll start with 13. So this is the one you guys did the OF glucose medium test and starch hydrolysis test. So the starch hydrolysis test is pretty easy. Just know that, let's see, what do they call the Petri dish? It's a special dish that has, it's a Petri plate containing the nutrient starch agar. Okay, so this test is to find out whether these bacteria metabolize starch. So all you're going to do with you're going to go get your Petri dish, get some iodine, and pour it on there. If the bacteria does metabolize starch, there's going to be a clear area, because what iodine does is it binds to the starch, and it kind of dyes it. So it's dyeing all this because there's no starch left in this area because the bacteria kind of broke it down, so it's not there anymore. If the whole plate is dark, it means that bacteria does not utilize starch. So he could potentially have something like this just sitting on the counter and say, does this bacteria metabolize starch? So if there's a clear area, the answer would be yes. That one's pretty straightforward. So just know the purpose of the iodine is to, it's kind of to stain the starch dark blue. That's the purpose of the iodine. Okay. So the OF glucose test is a little bit more, you have to read a little bit more into it. So if you guys remember, you did 2 tubes, and one of them, that's my tube. So one of them had mineral oil on the top, and one of them was just exposed to the air. So this one, the one that's exposed to the air, that is the aerobic tube, or if you get a reaction in this tube, if it turns yellow, that means your bacteria isn't, let's see, it's oxidative, because it needs oxygen. This is your anaerobic tube, so it has no access to air here. So if this one changes color, it's a fermentative tube. Okay. So if he has two tubes, and this one changes color, does this one oxidate glucose? Because these tubes, the thing is with these tubes is they have glucose in them, so you're testing to see whether the bacteria utilizes glucose. So do you guys know if this one would be, if this one changed color, what can you tell about the bacteria? It uses glucose, and also, since it has access to oxygen, it's oxidative. Okay, so what if just this, well actually, what if both of them change color? What could you tell about that bacteria? Yeah?
Doesn't that one use oxygen,
Yeah, so what you can tell for sure is it is definitely a fermentator. And it may or may not be an oxidator because fermentation can occur with or without oxygen. So you'd have to do further tests, but you would know for sure that it's a fermentator. But a pretty good indicator is that if it's an oxidator, it'll kind of just change color, like at the top, and at the bottom it'll remain the beginning color because it doesn't have as much oxygen down here. So I hope that wasn't too confusing. I hope I cleared it out. But if you go into your lab and this one changed color, this one didn't, it's definitely an oxidator. Just know that. So the OF in the title OF glucose, it stands for oxidative fermentation, fermentative, so because that's what you're testing for. So what's the purpose of the mineral oil in the OF glucose test?
Not let any air in.
Right, to block out the air, basically. Okay, and this is a little bit tricky. What if neither of the tubes turn yellow during the glucose test? Because these tubes, they contain glucose, so if they don't change color, what could you kind of?
Bacteria doesn't.
It doesn't use glucose at all. Yeah, it could use peptone or it could use some other type of energy. Anything else? I remember because Lisa and I, we were lab partners and we went over this
Like
a lot
20
with thing because both of our tubes turned yellow and we're like, so is it a fermentator or an oxidator? And he's like, well, it's definitely a fermentator, but you don't know if it's an oxidator. And so we're like,
Why do it? Just go to the other test.
yeah, so just know that if both of them turn, it's definitely this, may or may not be. And you'll go over it in lab today, so you'll actually get a hands-on experience. So it might be kind of confusing right now. Okay, lab 14 for me was the most challenging lab. When we did the review with Thane, he told us to understand each test, study one test at a time until you really understand it. You need to know what each test uses as energy and the byproducts of the test, of the fermentation or the oxidation that occurs. So in lab 14, so there's 3 different tests in lab 14. You need to know. Okay. And this is also one that you guys are going to look at today in lab. So you have the fermentation tubes. And this one's it's the carbohydrate fermentation test. So what you're just looking for is you want to see if this bacteria ferments carbohydrates, because that's what is in these tubes.